Thank you for participating in the NIA and NHLBI Entrepreneur Workshop Series. This workshop covers the topic of building your regulatory strategy. This breakout room, moderated by Francesca Mokel, discusses the topic of laboratory-developed tests.
Okay, great. Let's get started. So in the overview session, I gave a brief summary of what laboratory-developed tests are and how CMS under CLIA plays a very important role. So I wanted to see if there are any follow-up questions to what I said earlier or any detailed questions. Otherwise, I'll just see in the chat and in my advanced questions which came up. So let's get started then with a question that I touched on a little bit with regard to FDA's plan on oversight for LDTs. That's one question I saw come up, and it's a very good question. Over the years, there have been many different attempts at regulating LDTs under FDA. There were draft guidance documents that were issued. None of them got finalized. And as I mentioned, the VALID Act, and now what some people call the VALID Act of 2021, may give an indication of how FDA intends to oversee laboratory developed tests. So shifting the oversight more from a CLIA standpoint, CMS CLIA, to FDA, and how would that look like? And I should tell you in advance, I'm not an expert in the VALID Act, but I'll just give you my understanding of what I've read. So one aspect, as I understand it, that laboratory developed tests would be grandfathered in. So that means that as an LDT exists at the time, let's say this VALID Act would come to fruition, that LDT would be grandfathered in, and the complicator here is that you could not change your test after that. What would happen if you were to change your laboratory developed test under the VALID Act? So that is where the concept of in vitro diagnostics comes into play. So FDA, as it was laid out in the VALID Act of 2021, so that proposal intends to pull out in vitro diagnostics under medical devices and make it what I believe is called in vitro clinical tests. So make it its own category, and then assess those tests based on the risk level. And that is not just done from a technology standpoint, but also from an analytical standpoint, so the software component. So those are the high levels of the VALID Act. And I'm going to pause here, see if there are any follow-up questions that I may be able to answer. So please feel free, raise your hand, type it in the chat. No takers yet. Okay, let me see what other questions I saw here. Okay. One question that came in is how can an LDT designation from CMS aid in a 510K application to FDA? That's a great question. So first of all, it's very complicated, I know. Laboratory developed tests are the tests that are performed at a CLIA certified laboratory. So there is no designation you get per se. The question with regard to how an LDT can be used for approaching FDA to get an FDA approval for your diagnostic is a very good one, in that you can use the analytical validity that is required to validate your laboratory developed tests in a CLIA lab, and can build upon that built the clinical validity, which is not a requirement for laboratory developed tests, into your case that you would make to FDA. So you can use the information and the data you've gathered running an LDT. So again, that is a test that is developed in a single laboratory and is completed in a single laboratory, so it cannot be licensed out anywhere. You can use that as a use case, if you will, to establish the performance of your test and more.
You do have a hand raised, Francisca.
Oh, yes.
Maria,
Oh, I'm so, Maria, I'm so sorry. I didn't see that. Please.
Yeah. Hi, my name is Maria Grazia Ruocco. And my question is, we are very early in the development of our product. So we know it's an in vitro diagnostic, but it's not very clear whether it will fall, or maybe it's not clear to me, the difference between de novo or FDA approved test versus a laboratory LDT, right? So how to, and this of course also changes the strategy of, my early my early contact with the regulatory, but also it may change the pathway I want to choose, whether I understand better. So how I can, figure that out, the difference out. Thanks.
Great question. Thank you so much for raising that. So let me try to explain it in a hopefully simple way. I can't guarantee you that, but I'll do my best. So you can think of a laboratory developed test as a clinical test that you would offer to patients that you serve through that laboratory. So I don't know what your setup is, but let's take a clinical laboratory that is connected to a hospital or health system. So that laboratory could develop a test validated in their CLIA lab and validated based on CLIA standards, and then offer that test to the patients that are served by that hospital and by that laboratory. What you cannot do under this circumstance is say, okay, I have an amazing and great test. I have other laboratories who might be interested in performing this test. You cannot license it out to other laboratories. So other laboratories cannot perform your test. Other laboratories, if they had your intellectual property, if you will, if they knew how to perform your test, they could set up a similar test. That is perfectly fine, but you cannot license it out. So a requirement for an LDT is that you have a laboratory that is CLIA certified or CAP accredited. So oftentimes we've seen innovators coming up with amazing ideas, but they only have a research laboratory. So that would require them to transform, if you will, their research laboratory into a clinical laboratory. And there are consultants that can help with that. So that's sort of the laboratory developed realm. Now, if you're thinking about making this test widely available, having a manufacturer produce your test, and then different laboratories across the nation running this test. So that would be an IVD requiring FDA clearance or approval. Now, it would depend if that type of test has a what we call predicate device. And that predicate device, you have to answer a couple of questions. Does it have the same intended use? Is it similar to yours? And if that is the case, you would go down a 510K pathway. And if you're interested in that, my colleague Ben Behrman can answer very detailed questions in the other breakout room. If there is no what is so-called a predicate device, then you would go down the de novo route. So hopefully that gave you a little bit of a flavor of the different regulatory pathways. Any other questions?
You have a new question in the chat as well.
And
But Jean,
yeah, I have a question just following up on what you say about assay that we develop in a R&D lab, and how do we like transform it into like a CLIA certified CAP lab. And you mentioned that we cannot like license it out. So can we like work with a existing CLIA lab to actually validate the test that we develop, the assay that we develop in our R&D, and then validate it inside our CLIA lab and then go that route or not?
yeah, excellent question. And the way it would work is that you can't license your IP to the laboratory, but if they have the knowledge of how to set up your test, they can do that. And then it would be considered a laboratory developed test. So that means they know the analytes to put on, they have the appropriate machines to run this test, and it would give you the output that you had intended. So if you're working in partnership with that lab, then that is a route. But again, then only that lab can use that test. Hopefully that's clear. I know it can be kind of complicated.
Right, so that lab is the only one that can run that assay. We cannot go into another CLIA lab and do.
Yeah, it's like replicating your test, right? So technically you can't sell them a kit, right, to run it. Just because they have the CLIA designation, they have to replicate that entire test. And then it's a laboratory developed test. And they have to run it through the CLIA validation processes. I'll give you another example of an LDT, perhaps that makes a little bit more sense of what I'm talking about. So instead of creating your own laboratory developed test, so you have an innovative idea about a biomarker, etc., you could take an IVD, so an in vitro diagnostic, off the shelf, and then adjust it and add an analyte or adjust it into what now becomes a laboratory developed test. So the moment you change the IVD that has an FDA approval, it becomes a laboratory developed test and now can only be performed in that one laboratory.
I see. How about the sample collection kit? If we ship, because we need like human specimen to analyze the assay, is that fall into IVD if the collection kit is something that we develop it as well to preserve the cell quality in order to run the assay?
Excellent question. So with regard to sample collection kits, it depends on where it is used. So if you're using the sample collection kit in the doctor's office that is associated with your lab, so the doctor sends the sample back to your lab, that is not an issue. Generally, it would become an issue or more complicated if you're asking the individual to perform the test at home, meaning to get a sample at home and then send it back to the lab. Now we are under the umbrella of potentially direct to consumer testing. And that is where these special cases come into play. For low risk tests, usually LDTs are not low risk per FDA standards. There are some exclusions, but it depends on what kind of test it is. but it is highly likely if you were to have a home collection kit that FDA would need to review it.
Yeah, excellent. Just last question. I'm
Yes.
done here. Where can we find more information on the test validation inside CLIA? Like, what is the process of validating the test inside a CLIA lab?
Excellent question too. So CMS, if you Google, for instance, or use a web browser, CLIA, CMS CLIA, it should come up as it's a PDF, I think it looks like, and you get an understanding of the requirements. If I have time and can find it quickly, I'll put it in the chat too. But it gives you an understanding of CLIA. I want to say it's not an easy task, but it's not impossible. And there are experts in the field who can guide you through that process. And then that opens up the door for your laboratory to do any kind of clinical test. So that's a plus. All right. We had a question with regard to CAP or COLA. So let me just clarify. I've used the term CLIA certified and CAP accredited. CLIA certified is sort of the standard in getting a clinical lab up and running. So that's the minimum bar. There are alternatives to CLIA, and it also depends on where, in which state you set up your lab and perform these tests. So there are some exclusions, I believe New York is one of them, where there are CLIA exemptions in those states. So that is one thing to keep in mind. So CLIA is a general term and generally widely used to get certified to become a clinical laboratory. There are alternatives to CLIA, such as CAP, and there we call it CAP accredited. And I have seen that a lab gets first CLIA certified and then CAP accredited. From my clinical lab director standpoint, CAP is a step higher than CLIA. Hopefully that answers your question. So CLIA is just the general certification type. There are alternatives to it, and then there are also exclusions depending on which state you're in. All right. Did I miss any questions in the chat? I think I answered those pertaining to LDTs. Feel free to raise your hand. Feel free to just ask a question. Otherwise, I'll go down my list here. All right, there is one question about wanting to hear more about clinical utility study topics that support FDA clearance for Class 2 LDTs. So a couple of things here. LDTs, hopefully I described it in an understandable manner, are under FDA's enforcement discretion. So FDA does not actively review LDTs currently. But if you want to go down the pathway, moving from a laboratory developed test to getting FDA clearance, you would do that through the pathways that I described. And again, Ben Berman, if you have questions about that, my colleague Ben Berman in the other room can answer those. So those would either be the 510K route or the de novo, and you would get FDA clearance that way. And there is a reference about a Class 2 device. It really depends on the risk assessment, based on FDA's risk assessment, as to how they categorize your particular test to fall into Class 1 through 3, 3 being the highest risk. And what else can I tell you about that? Clinical utility studies. Okay, I mentioned it briefly just to reiterate. Clinical utility or validity is actually right now not a requirement under CLIA. Analytical validity is tested. So However, to make your case, clinical utility studies should be thought about when you're building out your strategy for FDA clearance. And again, Ben Berman, my colleague, can answer those questions in more detail. Hopefully that answered that subject, and I'm going to switch over to see if there are any raised hands. Chat windows. And just a little bit about the CLIA classifications under FDA. I mentioned in my summary in the beginning that laboratory developed tests are considered high complexity tests under FDA's CLIA designation. So that means that your CLIA laboratory would have to be set up as a high complexity laboratory. There are two others, low and then moderate complexity categorizations. And those are determined by FDA after review of your application and after your device as an IVD would enter the market. And I can answer any questions on that if there are any. Right? Yes, Maria.
Yeah. So my question is, the high complexity determined by the type of reaction you have to do, the reagent number of reagent, whether you need an expert to use the test? How is the high complexity defined?
Yes, so high complexity means that as the from a couple of standpoints. One, the test would have to be performed in the laboratory by laboratory staff. So it cannot be a lay person. It cannot just be a healthcare professional. It is complex so that, again, a laboratory staff member would have to perform the steps in getting from the sample to essentially a test result, right? So that is one component. And then there are also other considerations pertaining to the risk of the patient. So for instance, how can the test results be interpreted? Does it need to be interpreted by a laboratory professional, specifically the lab director or the medical director of that laboratory? So that is the case for high complexity tests, or are they easy to interpret and could be done by, let's say, a healthcare professional? And I won't get into layman's persons because that's not an LDT, but if you have questions on that, I'd be happy to answer those too. Okay, so just Maria, regarding your question, if you think about an LDT, the assumption here, it is a high complexity test. It needs to be performed in a high complexity laboratory. Now, again, if you look in the future, you want to submit your application to FDA, then FDA would make that determination. And it could be that it is a moderate risk, moderate complex test, I should say, or low complexity, but it really depends on your test.
I have another question if no one.
Sure. Go ahead.
Yeah. So I know that this is about like regulatory, but this is a question related to reimbursement. Is a CLIA lab in a specific state can only, adhere to the Medicare contractor of that state and not like a IVD where it. So the CLIA lab would be the entity that billed the insurance company, correct?
Yeah. Excellent question. So it would depend. Yeah, there are different models. the more general one would be that the lab would bill for the services performed there. I've seen models where the healthcare provider performs the billing, but let's not talk about the exception. So yeah, you're right. So there are a couple of things to take into account. One, again, it's a regulatory webinar, but I'll just give you a couple of pointers. One is to look at what's called CPT codes. I don't know if you're familiar with it. What is your test called? What would be the general test classification of that test in terms of it is a non-invasive prenatal test, for instance. So I would look, again, you can search for it, for that terminology of your test, and then CPT code. And then look what CPT codes come up, and then combine that with a search for coverage, what's called coverage determinations by payers. So those are the commercial payers, and you would get an understanding of what commercial payers are covering or are considering exceptions. So they wouldn't cover it, they would deem it experimental. And then under the CMS umbrella, it depends again on your type of test and where your laboratory is situated, and what the MAC is, the Medicare, I'm blanking on it, administrator. Yes. So it depends there. It is state specific as to who your MAC is, your administrator for Medicare and Medicaid, how they're covering your test. But if you have questions, I believe the information was shared for our seed team. We would be happy to set up a regulatory consult, a reimbursement / regulatory consult for you too. That is for anyone. If you have questions, we'd be happy to do that.
Oh, that's great. So I should follow up with the seed team and ask for that.
Absolutely. There was a question, a good one, regarding what is the regulatory path from LDT to a point of care assay. So I have seen, so point of care technically would be, I believe, usually a moderate complexity test deemed by FDA. So I have seen a couple of versions. So I've seen a point of care test being run under a high complexity laboratory, but that doesn't answer your question. So the pathway from laboratory developed test to point of care. Point of care describes the setting in which the test is performed. So it is not an FDA designation. So they wouldn't give you point of care designation. They would give you, for instance, CLIA waiver designation. So let me walk you through that. I talked about FDA's CLIA complexity categorizations, and I talked about high complexity for LDTs. For point of care, you would have to establish that a healthcare provider or the individual who is taking the sample and putting the sample in your device, and so performing the test and then reading the result, can do so in a way that would not add additional risk to the patient. So it would have to be simple enough for, let's say, a nurse, I'm just giving you an example, a school nurse to run. The other component to point of care testing is that it would have to be completed in a, still in a site that has a CLIA certificate, and it can be done at a site, it could be a school, it could be a health fair, that has a CLIA certificate of waiver. So that is where waived tests, so FDA waived tests from a complexity standpoint can be performed. So again, just to reiterate, it depends on how complex it is to perform the test from taking the sample, putting it into the machine and reading the result. So that would be what FDA would be looking at. Hopefully that answered your question. And if not, please feel free to pose a follow-up question in the chat or live. I think those are most of the questions that I have seen come in. There was a question regarding software regulated by FDA. So here I would again refer to my colleague Ben Berman. He would be an excellent source for those types of questions. Same for pros and cons of de novo. So that would be a good question for my colleague Ben Berman. Yeah. Any other thoughts, questions? Well, we are approaching time. If there aren't any other questions, I would again encourage you to fill out the feedback form. It's really helpful for us to, as presenters, to understand in terms of addressing your questions, the themes, were we clear enough? And again, please feel free to reach out to the seed office. So Chris Sasiela is who you heard speak from the seed office, and Ben and I support Chris Sasiela there, and we would be happy to set up any additional sessions just for you and your innovation to address regulatory and even reimbursement questions.
Thank you so much.
Absolutely. I will stay on for a couple more minutes if there are additional questions, but I hope you took away that laboratory developed tests are fall under the FDA enforcement discretion. So that is one thing. They are developed and performed only in the lab that created it. There is a pathway to move from an LDT to an FDA authorized test. That is certainly an option. It would require more data collection. And again, my colleague Ben Berman could answer questions there. And then lastly, I hope you took away that CLIA and setting up a laboratory under the CLIA certification or an alternative to it, as I described, are one of the requirements in setting up a successful laboratory developed test. All right. Yes, Maria.
Just very quick. So how difficult it is to move from the LDT to the FDA? Because our strategy is to have the test first validated within our hospital system and then move out from that. And as we collect more data samples, it's
Great.
something that one can envision to do first the LDT and then the FDA if it's obvious way to go.
It would really depend on your device. So I would encourage you to actually set up a consult with us, and then we can go into detail.
Thanks. Bye then.
Of course.
Bye-bye.
Yes. Again, I will stay on for a couple more minutes. If there are last minute questions, happy to answer those. Otherwise, thank you so much for your participation. Please remember to fill out the feedback form, and hopefully we will talk at some point soon, and best of luck to you.
Thank you. This is really helpful so far. Thanks so much.
Great. All right. Since there are no additional questions coming through, I think we are ready to close this room. So thank you again so much. It was wonderful hearing from you, and hopefully I was able to answer, if not all, some questions. And like I said, please stay in touch with us, reach out to us if you have additional questions specifically to your innovation, and best of luck to you. Thank you.